we're going to have a brief introduction on exactly what celiac disease is. Celiac, can anyone hear me? Okay, sorry. Celiac disease is a digestive disease that damages the small intestine and interferes with absorption of nutrients from food. When people with celiac disease consume gluten, such as wheat protein, their immune system responds by damaging the small intestine. Celiac disease is a genetic disorder. Only HLA, DQ2, DQ8 positive individuals are susceptible. Symptoms include diarrhea, weight loss, and malnutrition. Diagnosis for celiac disease is done by intestinal biopsy, histological grading, and the presence of serum autoantibodies. Treatment is by withdrawing gluten from the diet, gluten-free diet, or GFD. Celiac disease is considered an autoimmune disorder because the body's own immune system causes the changes that causes the disease.
Okay, what we have here is a picture of the gut lumen, and right here you have the lumen, and as you can see, the lumen is composed of folds, and on those folds are small finger-like folds called villi, and this is a bigger picture of the villi. And if you even look closer, there are other small folds on those folds. The reason why there's so many folds in the gut lumen is to increase surface area for better absorption. What happened with celiac disease is that since the body is attacking the gut lumen, it destroys the villi and the many different folds, so you don't have that much surface area and you're not able to absorb as much food and nutrients as you used to. Okay, I haven't been working on this project for about a year, so I'm a bit shady on this. This is the immunopathogenesis of celiac disease, and basically gluten is taken into the body, and because the body believes that gluten is so harmful, it changes the shape of gluten to these diamond-like shapes that become gliding peptide. Those peptides bind to the antigen-presenting cell, and what happens in the body is that if usually proteins and molecules in the body have to present the cells and the protein that they contain to antigen-presenting cells, and if it binds, it means that the protein is not supposed to be in your body. This happens a lot with cancer cells. When the cells presents the molecules that are present inside the cell, when it binds, the body then knows that it's not supposed to be there, and the cell is eliminated. So because it binds, this molecule is presented to LPCD4 positive and CD25, which is then passed on to the B cell, and the B cell releases anti-gliding, which destroys the gliding peptide.
This slide tells the objective of our project, and the objective was to identify subsets of regulatory T cells in the lamina propria in patients with celiac disease. Regulatory T cells, also known as suppressor T cells, are a specialized subpopulation of T cells that act to suppress activation of the immune system and thereby maintain immune system homeostasis and tolerance to self. They are characterized by having some of the following markers: intracellular FOXP3, TGF-beta, interleukin 10, CTLA4, and GITR. This is a list of some of our methods. The first method that we had was to isolate lamina propria lymphocytes, LPL, from the duodenal biopsies of 30 patients with celiac disease. Immunostaining for the following extracellular and intracellular markers: T cell markers, CD3, CDF, CD4, CD25, and CD124, 127, I'm sorry. Markers for regulatory suppressor cells: intracellular FOXP3 cells, intracellular TGF-beta, GITR, CTLA, and intracellular interleukin 10. The last method that we used was flow cytometry. which is a technique for counting, examining, and sorting microscopic particles such as cells suspended in a stream of fluid. I didn't see that yesterday.
This is a picture of a flow cytometer. We called ours FlowJo, was it?
Yeah, I think so.
I don't know why we call it FlowJo, but a flow cytometer, and basically this is what happens inside. The sample of cells is taken in, and the cells are hit by light source, so that we can see the cells on the sheet without showing later on. And basically, whatever we're interested in is sorted, and then the rest is discarded. Results. All right. The first graph, we have basically everything that was in the flow cytometer, and this right here is the sample that we're interested in. So to make this bigger, we have CD4 on your y-axis and CD3 on your x. And over here is a high concentration of CD3 and CD4. So in this concentration, we also have CD127 and CD25. Now, these graphs here are to find the concentration of intracellular FOXP3 in these samples. And the sample here is CD127 high and also CD25 high, which has a pretty high number of intracellular FOXP3. This is a low CD127 and a low CD25, which has a low concentration of intracellular FOXP3. And over here you have a high 127 but a really low CD125, and it has a moderate level of FOXP3. Basically everything here put on the table. 30 patients, we had 10 active, 10 on the gluten-free diet, and those were the patients that had celiac disease. And since there's no cure for celiac disease, what we can do is just tell these patients not to eat anything that contains gluten. So that pretty much cuts off anything, pasta, bread, anything that contains wheat. And these were the normal patients. And CD4 positive and CD25 and CD127 low had about a 68.7 and percent of FOXP3. And the people in the gluten-free diet had 16.6, and normal had 11.6, and so on for the rest.
Okay. Our conclusion for the project that we did last year. Regulatory T cells are present in the lamina propria layer of the gut of patients with active celiac disease, as evidenced by intracellular FOXP3 staining of various cell populations, which is what was shown in the graphs. Although there are no differences between the relative percentage of suppressor T cells phenotypes, such as CD25 positive and CD25, CD127 low, in active celiac disease cases versus treated or normal controls, FOXP3 expression was only observed in active patients. These are our references. We'd like to acknowledge Dr. Manavalin, Dr. Satbhattacharya, Harlem Children's Society staff. Shannon was one of the other people in the lab last year, and the Celiac Disease Center of Columbus University. Thank you.
Any questions? Yes, please.
What did you define as active patients?
Active, what was active patients?
We have to show direct symptoms of celiac disease.
Yeah, those are the patients that had celiac disease but were not yet put on the gluten-free diet. Okay. What'd you say?
Which one?
I think the flow cytometer, and I will also say the intestinal biopsies, because the pieces of intestines of the patients were given to us, and then we did the whole process. Yeah. Any other questions? Thank you.